Hello class, I'm now going to give you your lecture on hematology, which is the study of blood, and in this lecture I'm actually going to focus mostly on not just hematopoiesis, which is the production, synthesis of all the blood cells or fragments of cells that we associate in blood, but I'm going to focus on erythropoiesis and disorders of red blood cells. Erythropoiesis refers to the production or synthesis of red blood cells. You can't have erythropoiesis without hematopoiesis. So, the content that I'm going to cover today is in chapters 32 and 71, and this content will be delivered in about 4 segments. So it will be labeled Erythropoiesis Part 1, 2, 3, and 4. We have a lot to cover. I'm going to start out with a general overview of what we find in blood and then what we find in plasma, and then hematopoiesis, how we make all of our cells that are associated with our blood. Of course, that means red blood cells, all the white blood cells, and even platelets. And then I'll focus mostly on erythropoiesis. After that, I'll go on to our red blood cell indices, characteristics of red blood cells, everything that you need to know as future caregivers. Then I'm going to move on to how we produce red blood cells and maintain that value, and how we remove them from the circulation. After that, the discussion on normal production and removal, I'll talk about all of the red blood cells. Well, I shouldn't say all. I'm going to highlight a significant number of red blood cell pathologies. I'll talk about the different types of polycythemia, and then I will focus on quite a few of the anemias and how to classify them. Alright, so what is the function of blood? Well, blood transports many things. It transports our dissolved gases like oxygen and carbon dioxide, as well as anything else dissolved in the plasma compartment like nutrients, say for example free amino acids or fatty acids and glucose, hormones. It also carries away waste from the tissues of the body, and antibodies. It helps regulate the body temperature. Blood is important in maintaining the body's pH. It's also very important in maintaining the body's fluid volumes. We've learned about our different body fluid compartments. And blood is very important with helping to maintain our electrolyte levels. We also find white blood cells that are obviously very important in fighting against pathogens or antigens, things that are not of self and provoke our immune response. And as you will learn in another segment, in blood we find our platelets, which are involved in hemostasis. So what are some characteristics of blood? Well, the adult male has more blood than the adult female, typically. The adult male has about 5 to 6 liters, whereas the adult female has about 4 to 5. For our class, for testing purposes, we're going to pretend that we are unisex, and we're going to just stick to 5 liters. The temperature of our blood is about 100 degrees, 100.4 degrees Fahrenheit, and it is true that blood is thicker than water. It is 5 times more viscous than water. Now my question to you is, what actually makes the blood so viscous? And the answer is, the most abundant formed element in the blood is the reason why the blood is so viscous. You are 100 trillion cells large, and the most abundant cell in your body is the red blood cell. You have about 25 trillion red blood cells, and they are the reason why your blood is so viscous. Your blood pH ranges between 7.35 to 7.45, but as I told you in the first week class, our class normal value will be 7.40. We will not entertain a range, and this is for good reason. This is because when we start learning about acid-base balance, I want you to stick to a hard and fast number. So 7.40. What can happen at either extreme? We can have proteins denaturing. And I talk about the proteins and how they denature in our gene expression lectures. So please review that if you can't remember what denaturation is. The color of blood ranges. Oxygen-rich blood is very bright red, and oxygen-poor blood, in most textbook pictures, they draw it as if it's blue, and it's not blue. It's actually a dark crimson color. So oxygen-poor blood is still red, but it's very, very diluted in color. It's not bright and intense red. That bright, intense red color is only when there's oxygen around. So what do we find in whole blood? Whole blood, as you learned from your body fluid compartments, is actually a mixture of body fluid compartments. We have extracellular fluid, which is plasma, and then we have the formed elements, which are blood cells that are suspended in the plasma, and within these formed elements we find intracellular fluid. So plasma consists of 55% of the whole blood volume, whereas the formed elements are about 45%. And if you look at the yellow text box of the formed elements, the most abundant formed element, like I said in the previous slide, is the red blood cell, also known as the erythrocyte. The platelets or thrombocytes and the white blood cells, all of the white blood cells, the B cells, the T cells, eosinophils, basophils, macrophages, all of that consists of less than 0.1%. What about plasma? What components do we find in plasma? We find water. Greater than 90% of our plasma is water, followed by plasma proteins that are dissolved in this watery solution, and then other solutes like electrolytes, hormones, antibodies, etc. Well, antibodies are plasma proteins, sorry. So other solutes would be metabolites like fatty acids, amino acids, glucose, hormones that are steroid derived, anything that's not protein. So let's analyze this a little bit further. Water, like I said, is the most abundant component of plasma. Plasma is over 90% water, followed by plasma proteins, the most abundant of which is albumin, which is made by the liver. And you learned in your body fluid compartments that albumin is very important in helping to maintain your osmotic pressure in plasma. We also know that albumin can help transport different types of hormones that need carrier proteins. Although I must come clean on this, most of the hormones that need carrier proteins actually have their own designated carrier protein, and it's usually named as such. For example, cortisol carrying protein. So albumin can help transport hormones, but typically there is a different alternative plasma protein that helps carry most steroid hormones, and they're named after the steroid hormone they carry. Globulins like gamma globulin, alpha globulin, those are the other types of plasma proteins we find, followed by clotting proteins like fibrinogen, and you will learn more about fibrinogen in our thrombocyte lecture. And then all the other regulatory proteins like hormones, and there are even enzymes found in plasma. These enzymes typically chew up other proteins. That's why I said to you in one of our first lectures, what's one fate of of a hormone, and it might not necessarily reach its target tissue. It might be destroyed by a plasma enzyme. The other solutes that we find in plasma consist of electrolytes, organic nutrients like glucose, cholesterol, other lipids, amino acids, etc. We also find waste products like uric acid, which is the byproduct of nucleic acids, urea, which is the byproduct of protein metabolism, and creatinine, which is the byproduct of muscle metabolism, just to name a few. Now let's talk a brief moment on where we have hemopoietic activity. Where do we find our blood cells being made throughout the lifespan? Well, during the embryonic period, our blood is made in the yolk sac, and I need you to know this for the test. During fetal development, our blood is then made in the liver and spleen. And then shortly before birth, this hematopoietic activity is going to shift to the red marrow of our long bones in the hollow cavity, medullary cavity, and that you see up here as bone marrow. Now it says that this transitions to the bone marrow in the adult, but it's really shortly before birth, not actual adult status. And then you're seeing that around age 20, most of our hematopoiesis is occurring in our vertebrae and sternum, our axial skeleton, and we've lost virtually all of our red marrow in our long bones. And that red marrow in our long bones is being converted to yellow marrow, which is really just fat. So you oftentimes hear me say in class that after age 20 it's all downhill. So very early on in life we lose our appendicular skeleton red bone marrow, and now hematopoiesis only occurs in our axial skeleton, specifically the vertebrae, the vertebral bodies, and the sternum. So what is hematopoiesis? As you heard me say in our gene expressions lecture, hematopoiesis occurs or is possible through the mitotic divisions of a multipotent hematopoietic stem cell. This mama progenitor stem cell, as I like to call it, is called a hemocytoblast. And this cell replicates and divides through general mitosis that you learned in Bio 20. And when this mama progenitor stem cell replicates and divides, she produces two general colony or two general camps of cells. They're either the myeloid cells or the lymphoid cells. Now the lymphoid camp cluster of cells can only differentiate into T cells or B cells, lymphoid cells, lymphocytes. And T cells, of course, include cytotoxic T cells, helper T cells, etc. Now, the other general camp of cells that the hemocytoblast generates is the myeloid stem cell colony. And this colony of cells will be provoked, be pulled into different types of differentiation lineages. Let me explain that another way. So some of these cells might enter into the red blood cell lineage. Some might enter the basophil lineage or the megakaryocyte lineage to generate platelets. So each of the cells in the myeloid stem cell line will be exposed to growth factors and differentiation factors, and precursor cells will enter into these different lineages to turn into a very specialized kind of cell. It's sort of like saying the myeloid stem cell line is the stem cell line that will give birth to future nurses. And we all know that there are many different types of nurses. Are you going to be a telemetry nurse? Or are you going to be in labor and delivery? Or are you going to be an emergency room nurse or operating room nurse? You will become specialized. So what is the recipe for normal erythropoiesis? Obviously, we have to start with a very healthy stem cell line, the hemocytoblast line. And then all the daughter cells have to be pulled into their lineage, and they need to differentiate as they go through or progress through this lineage. So we need growth inducers, we need cytokines, we need differentiation inducers. These are all special chemicals released by adjacent cells or the interstitial environment that help promote the differentiation of the cells. We also need healthy cell division. We need our hemocytoblasts to replicate and divide appropriately and correctly. This means, of course, if we talk about the mitotic process, we know that there is, of course, an S1 and, not S1. We know that there is a G1, a growth 1, that followed by an S phase, followed by a G2 phase, followed by the M phase. And in the S phase, we have DNA replication. So we need, for DNA replication, vitamin B12 and folic acid. I need you to know this. Pay attention. In order to replicate your DNA correctly, so that a cell could divide, because the DNA synthesis phase is a precursor to the mitotic phase, you need coenzymes. And I'll explain what a coenzyme is. But coenzymes, vitamin B12 and folic acid, are extremely important for DNA synthesis. And that is a prelude to cell division. We also need ample amounts of nucleotides like adenine, thymine, cytosine, and guanine. And it turns out that vitamin B12 and folic acid are very important in the thymine pathway. Without vitamin B12 and folic acid, the cells, the precursor red blood cells, would not be able to divide. And without vitamin B12 and folic acid, we see individuals having very large red blood cells. They have fewer larger red blood cells instead of having many numerous smaller red blood cells. This is a type of anemia called pernicious anemia that I will talk about more in another segment. What else do we need for proper erythropoiesis, which is the synthesis of red blood cells? We need all the ingredients for hemoglobin synthesis, and I will talk about this more in our next segment. We need amino acids for the globin chains, we need iron, and we need the pyrrole rings. And the pyrrole rings are made in the mitochondria. I'll show you a picture of them. When we insert iron inside this ring, then we can call it heme. Now, for testing purposes, on the left side picture, or left side of this slide, you're going to see a picture of erythropoiesis. And I do not want you to memorize every step. I don't want you to worry or fret over a proerythroblast is the precursor to the basophil erythroblast, which is the precursor to the polychromatophil erythroblast. Oh my gosh, these are hard to say. You get the point. But what I do want you to know is that it is the reticulocyte that is released into the plasma from the bone marrow. And this is not a mature red blood cell. A reticulocyte still has endoplasmic reticulum, and it's the endoplasmic reticulum where we find some protein synthesis. So up until the very last moment, this cell is focusing on making hemoglobin, which is the most abundant protein inside a red blood cell. It's going to take several hours, a couple of days, for the reticulocyte to shed the remainder of this endoplasmic reticulum and fully differentiate into a red blood cell. So let's go through that again. If the mama progenitor stem cell, the hemocytoblast, which is a multipotent stem cell, which means it can replicate and divide and give birth to daughter cells, but the number of daughter cells it gives birth to is limited. So the hemocytoblast can only give birth to daughter cells that will ultimately differentiate into cells that we find in the blood. If we specifically talk about the myeloid stem cell line and specifically talk about the lineage for making red blood cells, then we are talking about erythropoiesis, and it takes about 5 to 7 days for a daughter cell coming from the hemocytoblast to fully differentiate into a red blood cell. And again, I don't want you to memorize the different stages, but I want you to know that as we go from the top of this picture down towards the bottom, the cells are becoming more determined, and they will ultimately towards the end be differentiated. But a hemocytoblast is determined. It is determined to give birth to daughter cells that can turn into types of blood cells, but a hemocytoblast is not a differentiated cell. As I said, the reticulocyte, if you notice towards the bottom, the reticulocyte is the cell that is released into the circulation. It shouldn't be more than 1% of all the circulating red blood cells. It has lost all of its organelles, including its nucleus, but it still holds on to some endoplasmic reticulum, which provides a platform to make hemoglobin. So up until the last moment, this cell is focusing on hemoglobin synthesis. Now, why would it be a good thing that mitochondria Why would it be a good thing that red blood cells lack mitochondria? Well, their purpose is that they are transporting oxygen. Oxygen is found dissolved in our plasma, but it is also transported in red blood cells, specifically bound to the iron centers of our hemoglobin molecules. So if red blood cells had mitochondria, mitochondria utilize oxygen. And if a red blood cell had mitochondria utilizing oxygen, then some of the oxygen the red blood cell would be carrying would be used, consumed, and therefore less oxygen would be available to the tissues of the body. So that's an advantage to the tissues of the body, that the red blood cell lacks mitochondria, but it's a disadvantage to the red blood cell because now that means it's going to have to use anaerobic mechanisms for glucose synthesis. It's got to use something else to make ATP. It can't use oxygen as the final electron acceptor in ATP synthesis. That oxygen has to go to the tissues of the body. So red blood cells are really taking one for the team, if you will. They are transporting oxygen, giving it to the rest of the tissues, but the red blood cells, they still need ATP. They still have cellular needs, and they are going to use anaerobic means to make ATP. And we will find out towards the end of this lecture that that can actually be very problematic for them. All right, on to segment number 2.